Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions. Your questions in the question and answer window will only be visible to myself and our speaker. Thank you for joining us today. It is now my pleasure to introduce our speaker, Kevin Lee from Erbi Biosystems.
Hello, everyone, and thank you for your interest in the Erbi Breeze microbioreactor perfusion system. Perfusion culture has many benefits, such as reduced footprint and increased productivity, but often it also increases the labor and complexity for operation. The extra hardware required can take up significant space. The volume of media required can be hundreds of liters a week, and maintenance can take significant time and effort with so many moving parts. At Erbi, we believe that going small can solve many of these issues. Our goal is to have one operator easily running 12 fully functional stirred tank equivalent bioreactors in the same 3-foot space of a single stirred tank. Erbi is about enabling rapid bioprocess innovation. We asked, what can we integrate all of the functions of a perfusion stirred tank into a single microfluidic package, and how small can we make it? By answering this question, we have created one of the only milliliter scale continuous perfusion bioreactors on the market, and our collaborators have been able to easily use our system to achieve high performance cultures beyond 200 million cells per milliliter. When we say easy to use, we mean that all required bottles, pumps, mixers, filters, tubing, and sensors are all integrated into the disposable, so there is zero assembly, sterilization, teardown, or cleanup. The small milliliter working volume also significantly reduces time spent on media and inoculation preparation, as well as in-process media refills and exchanges. At 2 ml, our provided bottles can run a 1 volume a day culture for more than a month without refill. We have demonstrated a variety of functions, including N-1 perfusion, media exchanges, and cell concentration, all performed with a simple software interface. In addition, there is no sharing of any functions, so each bioreactor can be run independently and inoculated asynchronously, which is great for testing different parameters. The ability to be representative of larger scale production systems is also very important. We will show you examples later of how we have been able to achieve this. Part of what makes our system so easy to use is our software. Users are guided through every step of the process, from calibration and inoculation to sampling and ending the experiment. Since all bioreactor control functions are integrated into the cassette, everything is controlled in one place. All online data and set points can also be visualized easily, and all measured process variables can also be plotted in real time. Sampling is also much simpler than other systems and is performed through software with the push of a button. Simply type in a desired sample volume and click go, and the system will automatically and sterilely eject a sample of any volume 20 microliters or larger. The key to the Breeze system is an integrated package of valves and pumps. By combining all of the fluid control in a single microfluidic package, not only is reliability and consistency improved, but complex functionality and control can be automated. Let's walk through the flow path to see how we provide continuous input and output flow. A fluid enters from one of 4 bottle inputs on the left and fills an integrated reservoir. The single microfluidic input pump downstream of the reservoirs can select which input to use. Since there's only one pump for the system, the cassette pump volume can be pre-calibrated at the factory before use. The pump injects fluid in the growth chamber, which consists of 3 chambers interconnected at the edges where the cells continually circulate. Periodically, the fluid in each chamber is expelled to generate flow into the 2 other sections, which provides mixing and gas transfer. There's also a cell retention filter integrated into the base of the growth chamber. This takes advantage of the chamber mixing to continuously clean the filter during operation. For fluid removal, the growth chamber can be pressurized and fluid selectively removed through the perfusion filter, the waste port, or a sampling port. So how does this type of device work? We use a thin silicone membrane to drive fluid motion by either deflecting it into a lower or upper chamber made in rigid plastic layers. This motion is driven by air pressure or vacuum. By properly designing the shapes of the cavities in the plastic layers, a variety of structures can be produced, such as valves, reservoirs, pumps, and mixers. Since the membranes are fully deflectable into the chambers, we have very well-defined volumes determined by the shape of the plastic and not by the flexible silicone. The cassette also has a variety of integrated optical sensors. There's a dissolved oxygen sensor embedded in the reactor that measures every 15 seconds. We also have two pH sensors on the system. A main pH sensor measures every 2 minutes and a second that measures once a day to correct for photobleaching. Headspace gassing coupled with the internal sensor measurements allows us to fully automate the calibration process for the DO and pH sensors. The process takes just a few hours and runs simultaneously for all reactors with a single click in software. We also have unique optical density sensors. Since we do not have bubbles, we can take cell density measurements exactly like a cuvette. This can significantly reduce the need to take out cell samples for density measurements. High-density cultures are possible using our mixer, which simultaneously moves the fluid and delivers gas. Silicone has a very high permeability to oxygen and CO2, so we can adjust the gas mixture in each of the sections to control oxygen, pH, and CO2 stripping, all through gas diffusion. This means no need to sparge bubbles or add antifoam since the system runs bubble-free. Since the mixer is able to completely fill or empty one of the growth chambers, a density measurement system is easily integrated. The flexible membrane allows us to configure the measurement chamber to either be full of cells or completely empty like a blank cuvette. By doing this, we can have optical density measurements that don't drift over time. There are two sensors of different path lengths in the growth chamber to provide linearity over different cell densities. At the bottom right, we have an example of one of our collaborators in the T-cell space, where they were able to demonstrate correlation of this sensor with their offline count. Many questions come up about how we sample in a system of this size. Most standard analysis equipment is actually able to handle our sample volumes, provided we do an additional dilution step. This dilution is generally required anyways at high density. For metabolite measurements, supernatant can also be spun down and diluted, and daily perfusion harvest volumes are more than enough to be measured directly. We have had the privilege of collaborating with Sanofi in Massachusetts to test our system against the representative stirred tank process. This plot shows average data from 4 independent Erbi cultures from an antibody-producing CHO cell line. The 4 cultures were quite consistent and able to meet customer expectations, as well as achieve cell densities greater than 140 million cells per milliliter with better than 90% viability. You can also see that in this case, there actually were not many cell samples taken. The customer was able to use the OD sensor as a proxy for culture growth and did not need to frequently verify with offline measurements. This cell line was also producing quite a bit of product, greater than 1 gram per liter, which met their expectations. We can also see that our unique silicone mixer is more than able to handle the oxygen demand at this cell density. The plot on the right shows both the measured dissolved oxygen as well as the amount of pure oxygen supplemented to be able to control. Since KLA is fixed in our system, the oxygen supplement is directly related to cell activity and is a unique feature of our system that can be used to gauge cell health and activity. Overall, the customer commented on the ease of setup and use of the system to achieve these results. Data here is based on a poster publication from Millipore Sigma using our system, and their standard process was performed in a 3-liter glass bioreactor. Their 3-liter process had 2 different cell lines and was compared against our Erbi Breeze reactor. And we also had one additional test comparing identical process controls in the 2 systems. The evaluations of the Breeze bioreactor process demonstrated reduced stress on the culture, resulting in improved viability compared to the standard 3-liter process. Further testing with identical process controls was able to demonstrate matched performance in the 2 systems under varied control conditions. The customer mentioned that the microbioreactor was better than other small-scale models due to the integrated cell retention device, continuous fluid flow, automated cell bleeding, and DO and pH control. We also had the benefit of collaborating with KTH to test the performance of the Erbi system. They were very interested in running perfusion experiments faster, so we tested high-density inoculations as a way of speeding up getting to steady state. You can see from the plot that they were able to consistently inoculate at over 20 million cells per milliliter and speed up what would normally be a week or longer growth phase to less than 4 days. This is especially useful for testing and optimizing media at high cell density perfusion processes. The small amount of media used also means that the Breeze system is uniquely positioned to test performance of high-value additives with very little cost. In addition to high-value media components, there are also cases where the cells themselves are high value. Erbi Biosystems has engaged with a cell therapy collaborator in their process development to scale to manufacturing by examining donor reproducibility through growth and phenotype in different media systems. Based on their initial experiments shown here, the collaborator has commented on overall ease of use of the system and great consistency between the pods, thus allowing them to test more conditions in less time. You can also see that they were able to test 2 different types of media using perfusion and duplicate on a single Breeze system. In the first panel, you will be able to see how closely the process can be managed across our bioreactors. Cells were grown in static culture through day 9 and then transferred to the Breeze for expansion. Growth rates were consistent among donor and media condition replicates. The second set of data indicates that not only is cell growth consistent among pods, but so are the phenotypes of the cells in the same conditions. It's important to highlight that for donor 1, the collaborator was able to detect differences in both cell growth rate and phenotype, depending on the media used. This suggests the Breeze would be well suited for DOE media optimization. Because of the small volume of cells required, typically only 1 to 2 million cells, the Breeze system allows you to use the same donor material and media across multiple bioreactors to better understand your process and controls with less of the resources in time, cells, media, and space needed by most other systems. So we've been able to show you capabilities of the Erbi Breeze microbioreactor through a few examples of different cell cultures. The system performs just like a stir tank bioreactor with a hollow fiber filter, whereas other scaled-down perfusion systems with daily media exchange may not be representative of larger scale reactors where you have true continuous perfusion flow. By going small, there are even advantages compared to a typical stirred tank perfusion system. The entire system, including the reactor, is integrated, pre-assembled, tested, and sterilized, greatly reducing experiment time and experiment failure, as well as reducing hands-on time. The smaller volume consumed reduces both cost and labor and enables access to testing higher cost experiments with expensive reagents, such as lentivirus transduction or even radio-labeled metabolite studies. Finally, smaller volume means less cells required for each experiment. In the case of mammalian processes, high-density inoculations in excess of 20 million cells per milliliter can be sourced straight from a small shake flask. And in T-cell culture processes, a single donor cell can be used for many process optimization experiments, eliminating the confounding factors related to donor variability. We believe that these advantages make our system incredibly unique and well-positioned to accelerate customer development cycles to get new drug products to market faster. Thank you for your time, and I'm happy to take any questions.
Okay. So the first question is, are demos currently available?
Can you hear me? Yep.
Yes.
So yeah, demos are currently available. We are actively demoing and selling our bioreactor system. You can go to erbibio.com, erbi-bio.com, and send us a note on our website, including the contact us part of our website to request a demo. During the pandemic, customers have also been able to easily receive the system and set it up via remote walkthrough and training. And we've trained customers using the system this way, and they were able to successfully run their experiments as well.
Okay. And what cell types have you worked with?
We've worked with quite a few now. We have grown various bacteria strains for antibiotics and biofuels, including E. coli and Rhodococcus and Clostridium. We've also cultured yeast strains such as Saccharomyces and Pichia. And in mammalian cells, we've now done CHO cells, HEC cells, sp20 hybridoma cells, and as well as T cells. And we believe this technology is very effective with the variety of cell types and look forward to working with other collaborators to help them learn more about their cell culture and processes.
Okay. And how does cell bleed work in the system?
So cell bleed is controlled by cell bleed is controlled in the system by equalizing the volume. So the cells are mixing in the system, and we inject some liquid all the time because we're running in perfusion. And when the cell density needs to be bled, instead of removing the liquid out through the perfusion filter, we actually remove the liquid out through the cell waste port. And that can be controlled either continuously with a fixed rate or as a closed loop control off of the output density sensors.
Okay. And can the system run several 2 milliliter bioreactors in parallel?
Yes, it can. So the system itself consists of 4 individual bioreactor pods. And so each system can run 4 in parallel, and they can all be run differently. If you want more than that, we just simply have more systems on the bench that you can run.
Okay. And what technology are you using for retaining cells?
Our cell retention is done with a membrane-based porous filter, and the pore size that we use now is 1.2 micron, and that is made out of polyether sulfone. And as far as cell retention, we see that the cell retention is fine, and it seems to behave like other filters that you would normally find when you're running hollow fibers with the APFs or TFFs.
Okay. Well, I'm seeing some questions come in, and just looking to see. How about, okay, is the system available in Asia or Europe?
Yes, it actually is. We are actively working in both China and in Europe. In China, we have a distributor who's helping us to run demos as far as to sales. And in Europe, we are currently direct with customers.
Okay. And what studies have you done on evaporative loss?
So that's a good question. We do have quite a bit of evaporation loss, as you can imagine, since the surface area to volume ratios are high. Luckily, our system is well defined, and the KLAs are very consistent. So we actually, what we do is we map out the evaporation loss based on KLA and mixing type, and we have the system automatically control direct water addition based upon the evaporation losses we assume are going to happen in the system.
Okay. So I think that's all the time we have for questions. If we didn't get to your question, they will all be forwarded to Kevin, and he can follow up with you individually. So thank you very much for joining us. And thank you to our audience. As a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Bye.